Thank you so much, Mrs. Sari Lutfia, as the moderator for the nice introductions. And I would also like to thank the committees of ICOHPS 2023 for the opportunity given to me to speak here and deliver my ideas, my research, and my insights through my presentation with the title of Unraveling the Genetic Maze, Exploring MecA Gene Polymorphism in Methicillin-Resistant Staphylococcus aureus, or MRSA. To begin my presentation, there are three major points that I will talk about today. And the first one is about Staphylococcus aureus. The second one is about MRSA, or Methicillin-Resistant Staphylococcus aureus. And the third one is about the analysis and, most importantly, the gene polymorphism of MRSA. Move on to the first point. I assume you all have known Staphylococcus aureus very well, but let me refresh you with the information about it. Staphylococcus aureus, that often abbreviated as S. aureus, is a gram-positive with a cocky or spherical shape like this, and tend to be arranged in clusters that appear as grape kind of bacteria. And it is commonly found in the environment and is part of normal flora of the skin and mucous membranes, particularly in the nose, throat, and the genitals areas. Staphylococcus aureus, also a versatile and a pathogen known for causing a wide range of clinical diseases like infection, and it produces a wide array of virulence factors like toxins, enzymes, and surface proteins, which contribute to its ability to cause disease. An infection caused by Staphylococcus aureus can range from minor, like skin infection, for example, impetigo, to more severe and life-threatening conditions such as pneumonia, endocarditis, and sepsis. Here are the examples of disease caused by Staphylococcus aureus. As you can see, there are bacteremia and also sepsis here. Bacteremia is a medical condition characterized by the presence of bacteria in the bloodstream. Normally, the bloodstream is a sterile environment, meaning it is free from bacteria or other microorganisms. However, in case of bacteremia, bacteria have entered the bloodstream and circulating throughout the body. It can lead to infection and become sepsis. When it has become sepsis, it is a life-threatening medical condition that occurs when the body's response to an infection becomes dysregulated, leading to a systemic inflammatory response, and it can result in organ dysfunction and failure. And if not treated promptly, sepsis can be fatal. Here we have pneumonia, which is an infection of respiratory system, and MRSA can cause severe form of pneumonia, and that poses significant challenges for treatment and become a life-threatening condition. The third disease is endocarditis. It is a rare but serious medical condition identified by inflammation or infection of the endocardium, which is the inner lining of the heart chamber or the heart valves. And it typically occurs when bacteria or other microorganisms enter the bloodstream and attach to the damaged areas of the heart lining or the heart valves. Endocarditis can trigger serious complication, and if left untreated, it also can be a life-threatening condition. And the last disease is called osteomyelitis. It is a serious and often painful infection of the bone, as you can see here. It can affect any bone in the body, but mostly it happens in the long bones like arms and also legs, and the spine and also the pelvis. This disease mostly be caused by the bacteria. It can cause significant damage to the affected bone and surrounding tissues if not treated rapidly. So, what are causing the spread of pathogenic Staphylococcus aureus? Here are some causes. The first one is colonization. Some individuals are carriers of Staphylococcus aureus and can unknowingly transmit the bacteria to others or develop infection themselves. And then we have the poor hygiene, the lack of proper hand washing and hygiene practice can lead to the transfer of the Staphylococcus aureus from contaminated surface or hands to the mucous membranes or open wounds. And then the next cause is uncover or open wound. Staphylococcus aureus definitely can enter the body through cuts, abrasion, surgical incision, or any break of the skin's territory. And it's a common cause of wound infection, including surgical site infection. And the next cause is the existence of medical devices in the body. Staphylococcus aureus infection can occur as a result of contaminated medical devices or implants such as a catheter, prosthetic joints, and a pacemaker in heart. And there also sharing personal items. Infection can spread more easily in settings like hospitals, nursing homes, military barracks, where people are in close proximity. And then the last cause is the procedures in hospitals. Invasive medical procedures like surgery, catheterization, or ventilator use can introduce Staphylococcus aureus into the bloodstream or deep tissues. And so Staphylococcus aureus strains can be or can be not resistant to penicillin as the part of beta-lactams antibiotics. And when a Staphylococcus aureus is resistant to methicillin or oxacillin, and it is called methicillin-resistant Staphylococcus aureus or MRSA. And there are two types of MRSA. And the first one is TA or community-associated MRSA. And the second one is HA or healthcare-associated MRSA. Okay, so the CA-MRSA infection typically happen outside the healthcare settings. They often manifested as skin and soft tissues infection, such as boils, abscesses, cellulitis, and impetigo. MRSA is often susceptible to a range of antibiotics, but resistance patterns can vary. And risk factors may include close skin-to-skin contact, sharing contaminated items like towels, razors, and participating in contact sports. Also, living in crowded or unsanitary conditions. And MRSA strains typically belong to different genetic lineage than healthcare-associated strains. While the healthcare-associated MRSA infection commonly happen in healthcare settings, such as hospitals and nursing homes, they can cause more severe infection, including surgical site infection, bloodstream infection like bacteremia, pneumonia, and urinary tract infection. MRSA strains often exhibit resistance to multiple antibiotics, and this infection can be severe and are often associated with longer hospital stays. So, MRSA can be considered as a major public health concern due to its high infection rates, because MRSA infections are rapid, and it also has high mortality rates an invasive MRSA infection, especially those associated with the healthcare settings or HA-MRSA, have been linked to higher mortality rates compared to the CA-MRSA. And MRSA also very easy to be transmitted. And MRSA can spread easily from person to person through direct contact or touching contaminated surface. And MRSA also has challenges in treatment and preventing strategies because the infection of MRSA require alternative antibiotics and treatment can be more complex, difficult, and costly. Let me show you the example case of MRSA infection. And as you can see here, a New York Giants football player was hospitalized with a bad infection in his foot because of MRSA. And the infection reacted none to antibiotics, and he needed to go through several surgeries, and he never able to play football again. And then this is just the same. An athlete named Edward had MRSA infection, but fortunately in time, so in time for the diagnosis, so the doctor didn't need to amputate his leg. But in 2003, MRSA infection had killed Ricky Lanetti, a college football player, because of the pneumonia. Normal Staphylococcus aureus will produce an enzyme called PBP or penicillin binding protein that will bind to penicillin, and it will stop the Staphylococcus aureus from building its cell wall. But with the MRSA, there is a genetic mutation within the Staphylococcus aureus, and these genetic changes lead to the presence of the MecA gene. It is located within the SCC-MEC chromosome of S. aureus and encodes a specific transpeptidase and produce PB2A or penicillin binding protein 2A. This protein or enzyme has low affinity toward beta-lactam antibiotics like penicillin and methicillin. And PBP2A is not bound well with beta-lactam antibiotics, and this will allow the Staphylococcus aureus to continue building its cell wall instead of broke apart. So, how we can identify MRSA? Well, identifying MRSA in medical laboratory typically utilize conventional methods like oxacillin disc methods, and it involves a series of laboratory tests and procedures. And it requires pour, isolate, and also bacteria cultures. And the result of this method will be available approximately in 5 days. These methods detect the Staphylococcus aureus itself. Well, the more modern and accurate methods for identifying MRSA is by using PCR. PCR is not only for COVID identifying. And with this PCR, we need a bacterial culture, phenotype identification, isolation, and extraction of bacteria's DNA, and seal with PCR analysis. Here we can detect the MecA gene of MRSA. Currently, I and my team have been doing some research about the MRSA, and we've got the MRSA samples from RSU Haji Surabaya. In my research, we do the phenotype identification of MRSA, genotype identification of MRSA, MecA gene of MRSA sequencing, and also MecA gene of MRSA polymorphism analysis. We have done several analyses to identify the MRSA phenotypically, and the analyses were hemolysis, mannitol fermentation, gold yellow pigment, gram-positive, catalase, coagulase, oxidase, and urea breakdown, also sugars analysis and methicillin resistant sensitivity. From all the samples that we've got, we found that they all are identified as MRSA phenotypically. Based on the phenotype identification result, we continued to do DNA analysis, and this was begun with the DNA quantification. This is the process of determining the concentration of amount DNA in a given sample. Several kind of DNA quantification methods are available, but we use the spectrophotometry methods, and this is one of the most common methods. It measures the absorbance of the DNA on some wavelength. There were two wavelengths that were A 2060 and A 2080, and the ratio of A 2060 to A 2080 show how good is the DNA quality. As you can see here, we have the result that range between 1.8 to 2.0, all the samples. And it showed that all samples had good DNA quality. Next, we did the MecA gene optimization. And optimizing the MecA gene is typically involves the adjusting experimental condition to achieve the best result. And the goal of this procedure is to seek for optimal annealing temperature. We use MRSA positive control with MecA gene, and we have set the annealing temperature to be varied from 52.0 Celsius degree to 52.3 Celsius degree. We use the forward primer and reverse primer as shown, and we did the optimization in 30 cycles with the denaturation phase at 94 Celsius degree for 30 seconds, the annealing for 30 seconds, and the extension at 72 Celsius degree for 1 minute. So this is the result of MecA gene optimization on my research, that there was a strong band at 52.0 Celsius degree. And we have to select the annealing temperature that results in the balance between specificity and yield, and it will produce a band with a non-specific or smearing band. And we found that the optimal annealing temperature was at 52.0 Celsius degree. The next step that we've done was the MecA gene amplification. It was to make multiple copies of the segment, and this procedure were accomplished by doing the DNA denaturation, and then primer annealing, DNA extension, and we did the 3 steps in 30 cycles. And then we did the final extension and cooling to stop the reaction. And here we can see the result. There were 3 samples with no CT value. And it means that those 3 samples tested negative for the MecA gene, while the other 27 samples were detected positive, having the MecA gene. This negative result for the MecA gene in MRSA bacteria was suspected to be due to mutation or alteration in the DNA sequence of the MecA gene, rendering it undetectable using the primers employed. With this result, we have to do the polymorphism analysis of MecA gene. So, what is polymorphism? Particularly, what is MecA gene polymorphism? Well, it's a variation, genetic variation, or differences in the MecA gene that can exist among different strains or isolates of MRSA. And the goal of polymorphism analysis of MecA gene is to understand the epidemiology and evolution of MRSA strains, as it helps identify different genes. And there are 4 types of polymorphism analysis of MecA gene that are RFLP. RFLP is a molecular biology technique used to detect the variation in DNA sequences, and it involves digesting DNA samples with restrictions enzymes which cut DNA at specific recognition sequences. The second one is VNTR, or variable number of tandem repeats. And VNTRs are regions in the DNA where a short DNA sequence is repeated in tandem. And the number of repeats varies among individuals. The third one is SSR, simple sequence repeats. Simple sequence repeats are short, repeated DNA sequence, usually composed of 2 to 6 base pairs, repeated in tandem. And the last one is SNP, or single nucleotide polymorphism, which SNP are single nucleotide variation at specific position in the genome where one base, for example, A, T, C, or G, is replaced by another. The knowledge about gene polymorphism, especially the MecA gene polymorphism, including how to analyze it, is important to be studied because of these several reasons. The first one is we can understand the antibiotic resistant mechanism because the MecA gene polymorphism analysis will help researchers to understand the genetic variation responsible for the antibiotic resistance in MRSA. And then we can do the epidemiological tracking by studying the polymorphism of the MecA gene. Researchers can track the spread and the transmission of MRSA strains in healthcare settings and communities. This information is essential for epidemiological investigation and infection control measure. And then it is also important for clinical management because identifying specific MecA gene polymorphism can assist clinicians in predicting the resistant profiles of MRSA strains. This knowledge will help to guide the treatment decision, ensuring patients to receive the most effective antibiotics. And the fourth reason is for the surveillance. Monitoring changes in MecA gene polymorphism over time allows for the early detection of emerging antibiotic-resistant strains of MRSA. And this surveillance can help public health agency and healthcare facilities respond proactively. And then we can use it for vaccine and drug development because understanding the MecA gene polymorphism can inform the development of new antibiotics or therapeutic approach that target specific genetic variants associated with resistance. And then it also has the infection or prevention control or the infection prevention or control, because the knowledge of MecA gene polymorphism can inform infection control strategies in healthcare settings like hospitals, and it helps identify high risk of strains and implement measures to prevent their spread. And then we use it for public health planning. Information about MecA gene polymorphism is critical for public health planning and response to antibiotic-resistant infection. It helps prioritize the resources and intervention that the government should do. And the last one is for evolutionary studies, because polymorphism analysis of the MecA gene contributes to the genomic characterization of MRSA strains. And this whole reason is going toward to one big goal, that is to control MRSA infection and protect public health. Next, I want to explain quickly about the general steps of the polymorphism of MecA gene. The first step, we have a sample collection and DNA extraction. This step typically involves cell lysis, protein removal, and DNA purification. And next, we have PCR amplification. We perform PCR using specific primers designed for the MecA gene. PCR amplifies the target gene sequence. And then we do the DNA sequencing And here we sequence the PCR product, and this will provide the actual DNA sequence of the MecA gene in the sample strain. And the next step is a genetic variation or sequence alignment. Here we align the obtained sequence with a reference MecA gene sequence to identify any genetic variation or polymorphism. And then we do the polymorphism detection. Here we analyze the aligned sequence to identify variation such as SNP or single nucleotide polymorphism and others. And this variation are indicative of gene polymorphism. And then the next step is phylogenetic analysis. We construct a phylogenetic tree using the MecA gene sequence to understand the genetic relatedness among different MRSA strains. Usually, we use a computer application for this step. And the next one is genotyping. Here, we assign genotypes or profiles to MRSA strains based on their MecA gene polymorphism. This helps in strain typing and epidemiological studies. And the last one is we do the statistical analysis and interpretation. Here we perform statistical analysis to assess the significance of the observed polymorphism and their association with factors like antibiotic resistance or virulence. On my research, I will use the FFLP type of gene polymorphism to analyze the samples that I have. You've already knew that the result of the samples, the 30 samples that we've got, were 3 samples tested negative for MecA gene. So here are the steps that I will be doing. The first one is sample collection, and then DNA extraction, and then primer design. This is the important step of RFLP, the enzyme digestion. Here, we select the restriction enzymes that have recognition sites within the amplified region, and the digestion should produce fragments of varying length based on the presence or absence of restriction sites due to polymorphism. And the digestions with a restriction enzyme will cut the DNA at the specific recognition sites. And then we will do the electrophoresis separation and PCR reaction, then electrophoresis separation again, staining and visualization, and then interpretation or phylogenetic analysis with the phylogenetic software. Unfortunately, we haven't completed our research yet, but here I can show you the example result of the RFLP analysis of Ibrahim et al. In 2019, and they found that from their samples, there were 3 different patterns of coagulase length. That the first pattern show the 790 BP coagulase length, the second pattern show the 680 BP coagulase length, and the third pattern show the non-coagulase length. And they said that our study showed that the predominant genotype in patients at Kosti Teaching Hospital was CoA3, and 75.4% of CoA3 isolates were methicillin resistant. It could be presumed that the majority of infection in particular regions are caused by strains with certain CoA genotypes. And given its good discriminatory power, easy of use, and cost effectiveness, RFLP of the CoA gene can be used in epidemiological research to control and monitor hospital and community acquired infection. And now we have come to the summary of my presentation. That is, studying MecA polymorphism holds paramount importance in our ongoing battle against antibiotic-resistant bacteria, particularly MRSA. And ultimately, understanding MecA polymorphism is a powerful tool in our arsenal to safeguard public health against antibiotic resistance. Thank you so much for your kind attention.